I'd just like to begin by acknowledging the traditional owners of the land, the Wurundjeri people of the Kulin Nation, and to pay my respects to their elders past and present. Now, it gives me great pleasure to introduce Tan to you this afternoon. 3.5 years ago, Tan made the, I think, probably brave decision to commence a PhD under my supervision. I say brave because I'd never supervised a PhD student before, and I think it was quite a bit of a leap of faith for Tan to come on board, but I'm certainly glad that he did, because in the past 3.5 years he's shown himself to be a really stellar PhD student, and just as importantly, just a great person to have around the lab. Tan has a real gift, I think, for communicating his science, which I'm sure you'll appreciate over the next sort of 45 minutes or so. And this has been witnessed by a number of awards over the past 3.5 years, including Young Investigator of the Year Award from the Victorian Infection Immunity Network, as well as the Best Student presentation at IGV last year. More recently, Tan, with a travel scholarship from the Keystone Symposium, presented his work in Dresden, and he'll be talking about that work shortly. So just to finish then, I'd just like to say thanks to Tan for making my first experience as a PhD supervisor such a positive one. You've been great to have around, as I said, and I'd like to invite you up here to tell us about your work.
Thanks for that, Ken, for the kind introduction, and thank you to all of you guys for coming along and listening to my talk, which is looking at a relatively unknown or uncharacterized protein known as Sidt2 in its innate immune in its function or role in the innate immune response to double-stranded RNA. So making this talk over the last few days, I actually did something that people don't generally encourage scientists to do, which is to wait for a final result a few days before the talk. So I was doing that leading up to the weekend, and I didn't know which way this result would go, so I kind of made two presentations either side of that result. And it's not generally good practice, and I wouldn't encourage anyone else to do it, but luckily it was a very good result and a really significantly positive one, so I thought I'd just show it to you today. It was quite clear and quite significant, so I wish all my following experiments have been just as nice. That was the part I was most nervous about, and I think it's good. The rest of the talk's going to be fine. So, as I mentioned, I'm looking at a protein known as Sidt2. Sidt2 is one of two mammalian orthologs of a protein known as Sidt1 that is found, that was originally characterized in C. elegans, a nematode. And it was in these two seminal papers in Science by Craig Hunter's lab at Harvard, where they identified Sidt1 as an important gene and protein that was involved in the systemic RNAi phenomenon, I guess, in C. elegans, and it was they later characterized Sidt1 as a transmembrane protein that was able to transport double-stranded RNAs across cell membranes. So this was the experiment. Now that I'm thinking back when making these slides, I got a bit nostalgic, and this was an experiment from this original paper that really hooked me on Sidt1 and Sidt2. So I'd like to share that with you today by way of background. So what they did in this paper was to take a C. elegans worm, and they made a transgenic strain of this worm that expressed GFP within the cells of the pharynx, the muscle cells of the pharynx, and the body muscle down here. It's denoted by green. Then what they did was to express another transgene within the pharynx that produced a GFP hairpin. So the hairpin would overexpress this GFP double-stranded RNA, which would lead to knockdown through the RNAi machinery. So what they found was when they did that, when they overexpressed the hairpin within the pharynx muscles, they saw that it knocked down the GFP within the pharynx, but it also knocked down the GFP within the body muscle, which was driven over through a second transgene. So they're separate promoters. So that's what they called systemic RNAi. Then they did a screen, and they identified a mutant, or the first mutant they identified, where you expressed the transgene within the pharynx and you saw knockdown within the pharynx cells, but you didn't see this propagation or systemic RNAi. So they denoted this mutant as a Sidt1 mutant, or systemic interference deficient 1. Given that I was always disgusted and grossed out by worms, even as a kid, I was really lucky that Sidt1 was highly conserved throughout evolution, and mammals and humans actually have two orthologs known as Sidt1 and Sidt2. So this became the focus of my PhD project. So looking at the expression patterns of both Sidt1 and Sidt2, you can see that Sidt1 on the left column is relatively restricted in its expression. It's mainly found in glandular tissues and T and B cells, whereas Sidt2 is the more broadly expressed of the two orthologs, mainly in most organs here and most cell types. So we thought Sidt2 was the better target to look for. So the overarching question of my PhD was, what is the physiological role of the mammalian Sidt1 orthologs? So I was conducting experiments with Sidt1 and Sidt2 in tandem, but then towards the end, I've been mainly focusing on the second ortholog, Sidt2. So in mammals, the main role of double-stranded RNA would be in innate immunity, in acting as a potent and pathogen-associated pattern or PAMP, or danger signal to the immune system. It's mainly produced in the context of viral infections, where the virus is replicating within the cytoplasm of cells and producing these double-stranded RNAs. And these cells that will inevitably die, either by a lytic infection or by cell-mediated, immune system-mediated death, will release these double-stranded RNAs into the extracellular space. And these RNAs can be then detected by the immune system and lead to the production of type 1 interferons. And interestingly, it's not just double-stranded RNA viruses that produce double-stranded RNA during its life cycle. Double-stranded DNA viruses such as HSV also produce large quantities of double-stranded RNA, as well as positive-sense single-stranded RNAs such as EMCV. But its negative-sense single-stranded RNA viruses such as LCMV do not produce large amounts of double-stranded RNA. So once these RNAs are released into the extracellular space, they're taken up by neighboring cells by endocytosis and make their way through the endocytic pathway as shown here. And it's within the later stages of the endocytic pathway where they interact with TLR3 within the late endosomes and lysosomes. And this leads to downstream signaling through the transcription factor IRF3, which translocates and dimerizes in the nucleus to promote the transcription of interferon alpha and interferon beta. Alternatively, if a cell is already infected with virus within the cytoplasm, these viruses will replicate and produce double-stranded RNAs, and it's the role of the RIG-I-like receptors, namely RIG-I and MDA5, that sense these double-stranded RNAs within the cytoplasm and work through their adaptor protein MABs, which aggregate along the mitochondrial membrane upon activation to lead to IRF3 translocation into the nucleus, and again, the transcription of type 1 interferons. So what is slightly interesting and unknown property was that extracellular double-stranded RNAs can also be sensed by RIG-I and MDA5, suggesting that there was a way that these double-stranded RNAs, once being taken up by the endocytic pathway, can somehow escape from these endosomes and stimulate the cytosolic receptors. But the mechanism by which this occurs was previously unknown. Another interesting result was that MDA5 seems to be more important than TLR3 in the response to extracellular double-stranded RNA. And this was demonstrated in a paper in 2006 in PNAS, where they took BMDCs and treated them with poly-IC, which is a synthetic double-stranded RNA analog. So traditionally, when you wanted to study the role of the cytosolic double-stranded RNA receptors, you would complex your RNA or poly-IC with a transfection reagent such as lipofectamine or fugine, and this helps the cell take up the RNAs into the cytoplasm to stimulate the cytosolic receptors. And that's what they saw here. So wild-type cells, when you transfect poly-IC, they produce type 1 interferon, interferon beta in this example. TLR3, which is the endosomal receptor, TLR3 knockout cells are still able to produce type 1 interferon because you're bypassing the endosomes. But if you knock out MDA5, which is the cytosolic receptor for poly-IC, these cells do not make interferon beta. However, when this group just added poly-IC into the extracellular media and let the cells take up the RNA by endocytosis passively, what they saw was that wild-type cells, again, are still able to make interferon beta, albeit a little less. but so are TLR3 knockout cells, suggesting that there was a mechanism by which these cells that do not have the endosomal receptor are still able to signal to produce type 1 interferon. And again, when they looked at MDA5 knockout cells, they saw that these cells weren't able to produce interferon beta. This also translated in vivo, where they simply injected poly-IC into the peritoneal cavity of mice and measured interferon beta within the serum. So you can see here with TLR3 knockout mice, they're still able to produce type 1 interferon, similar to wild type, whereas MDA5 knockout mice were not able to produce interferon beta. And in this paper, they speculated that this was due to the presence of a transmembrane transporter or channel that may be mimicking the role of transfection reagents that we use in vitro. This wouldn't be that surprising, since the similar endosomal escape mechanisms have been identified in nature, literally in nature. So this letter that was published in 2014 identified two peptide transporters known as SLC15A3 and SLC15A4 as being endosomally bound, but they were able to co-localize with a bacterial peptide known as MDP. And when you overexpress these transporters, you saw an increase in NF-kappa B activation in response to either bacterial infections or MDP treatment, as demonstrated here by the black lines. And when they read out IL-6, they see, again, if you overexpress these transporters, you see increased IL-6 production, suggesting that these transporters are important in the endosomal escape of MDP or bacterial peptides from the endosome into the cytosol to hit NOD2 signaling. So we wanted to know, is Sidt2 our candidate for a very similar pathway, but with double-stranded RNA? So is Sidt2 responsible for the endosomal escape of double-stranded RNA into the cytoplasm for activation of RIG-I and MDA5? So the first question we had to ask, obviously, was where does Sidt2 localize? Is it within the endosomes? So what we did was to take mouse embryo fibroblasts that transiently expressed different markers of the endocytic pathway. So we have recycling endosomes, early endosomes, late endosomes, and lysosomes. And then we stably express Sidt2 tagged to an M-cherry, so we could actually look at its localization. And we can see here, when you merge the two images together, you see that Sidt2 predominantly localizes within late endosomes as well as lysosomes. Secondly, we showed that Sidt2 was also able to co-localize with poly-IC and as well as double-stranded DNA. So we see here DC2.4 cells that have been treated with poly-IC fluorescein, as well as double-stranded DNA fluorescein, so just a fluorescently tagged nucleic acid. And then when you overlay that with Sidt2 M-cherry, you can see nice co-localization with both RNA and DNA. So the next question we had was, does Sidt2 still retain its specificity or double-stranded RNA transporter function from Sidt1? So to identify this, or to look at this, we used fluorescence resonance energy transfer and fluorescence lifetime imaging, or FRET-FLIM. So I'll just quickly explain the principles behind the technique for people who don't know it. In these experiments, you have a fluorescently tagged donor and a fluorescently tagged acceptor as your either your ligand and your protein of interest, as I'll show you soon. So what you do is excite your donor cell or donor fluorophore and let it emit at a different wavelength that could be taken up by your acceptor. So if these two donor and acceptor molecules are close enough together, your donor should be able to donate the energy after excitation to the acceptor, and you should therefore see a reduction in fluorescence lifetime, as demonstrated here by the quenching of the green line. So in these experiments, we've replaced as our donor, we've used fluorescein poly-IC or double-stranded DNA, and as our acceptor, we're using Sidt2 M-cherry. So if these two are close enough together, you should see a reduction in fluorescence lifetime. And that's exactly what we see. So when we treat cells with poly-IC in the presence of Sidt2, you get this reduction in fluorescence lifetime, demonstrating that they are close together. Whereas if you do the same experiments, even though they co-localize with double-stranded DNA, you do not see this reduction in fluorescence lifetime, suggesting that like Sidt1, Sidt2 is also able to distinguish between double-stranded RNA and double-stranded DNA. Around the same time we got these results, there was another paper that was published showing that Sidt2 was also able to bind double-stranded RNA, and they showed that it was binding at a length-dependent manner. So Sidt2 seemed to be preferentially binding longer double-stranded RNAs compared to short, and this is very similar to what we see with Sidt1, so in C. elegans. So the next question we had was, what does Sidt2 play a role in the innate immune response to viral infections? And we generated knockout mice for Sidt2, and we put them through various viral models available at the Institute and at other institutes. So the first one I'd like to present today is the EMCV virus with the help of the Hudson Institute. We chose this virus because it was a positive sense single-stranded RNA virus that produces large amounts of double-stranded RNA, and I should mention that it has been shown previously that EMCV RNA is a main ligand for MDA5, so the cytosolic receptor. And also being a lytic infection, we figured this would be working in our favour, as cells will be lysing upon infection and releasing their double-stranded RNAs into the extracellular space. So when we infected our knockout mice, we see that mice lacking Sidt2 succumb to infection. Almost 100% of these mice will succumb to infection, whereas wild-type mice are able to survive through the infection. When we look at viral load within the peritoneal exudate cells, so this was an infection within the peritoneal cavity, so we just flushed out the cells within the peritoneal cavity and measured viral titers, and we saw that the knockout mice have increased levels of virus compared to wild-type, and given that EMCV has tropism towards the heart, we also see, we looked there and we saw, again, an increase in viral load compared to wild-type. When we measured interferon beta in both the PECs and the serum, we saw that the Sidt2 knockout mice produced significantly less interferon beta compared to wild-type controls. And then we did bioplex as well to look at other cytokines, and we saw a significant decrease in both IL-6, IL-12, and RANTES, which are pro-inflammatory cytokines. The next virus that we used was HSV1, or herpes simplex virus 1. This time, this is a double-stranded DNA virus that again produces a lot of double-stranded RNA and is a lytic infection as well, which we think is important. So when we look in vitro and we just infect wild-type and Sidt2 knockout MEFs with a fluorescently tagged HSV, we see that the knockout mice have higher rates of infection compared to wild-type. In vivo, around 50% of our Sidt2 knockout mice will succumb to infection around 3 days, whereas wild-type mice remain predominantly asymptomatic. And when we look at cytokines, again, we see a decrease in interferon beta, as well as the other pro-inflammatory cytokines, as we saw before. However, when we infected the mice with LCMV, which is a negative sense single-stranded RNA virus, that, as I mentioned earlier, does not produce large amounts of double-stranded RNA and is a latent infection, we saw no difference in interferon beta production within these Sidt2 knockout mice, suggesting that Sidt2 is only important for viruses that are producing large amounts of double-stranded RNA. So just to conclude the first part of my talk, I've shown you that Sidt2 localizes towards late endosomes as well as lysosomes. Sidt2 selectively interacts with double-stranded RNA, but not double-stranded DNA, similar to Sid1. And then Sidt2 knockout mice have higher mortality and produce less type 1 interferon in response to EMCV and HSV infection, but not LCMV infection. So after we saw this, we wanted to look at the actual mechanism of Sidt2 transport. So we saw a decrease in interferon beta and other cytokines, and we wanted to know, is this simply just because Sidt2 knockout cells have an impairment in their ability to take up double-stranded RNAs, and that's why you see less interferon beta. Given its localization towards endosomes, we didn't think this would be the case, but we thought we'd try it anyway. So we took bone marrow from wild-type and Sidt2 knockout mice and then differentiated them into dendritic cells. And then we treated them with either poly-IC rhodamine, so a fluorescently tagged poly-IC, or 32P labeled radioactive double-stranded RNA. And we let them take it up for an hour through endocytosis, and then we measured them 2 different ways. Firstly, with the poly-IC rhodamine, we looked at rhodamine signal through flow cytometry. And with the radioactive labeled double-stranded RNA, we measured the radioactivity as a surrogate for uptake. And what you can see in both cases is that there is no defect in Sidt2 knockout cells to take up double-stranded RNA or poly-IC compared to wild-type controls. So we treated the cells in 4 degrees as a negative control, where you would not see any endocytosis, and that's what you see. And at 37 degrees, both wild-type and Sidt2 knockout cells can take up the same amount of poly-IC. And that's demonstrated here in the radioactive labeled assay as well. So we concluded that Sidt2 does not play a role in the initial internalization of extracellular double-stranded RNA, and any defects we see is probably due to an intracellular transport defect. So what we then looked at the intracellular staining of double-stranded RNA or poly-IC within these cells. We took dendritic cells, treated them with poly-IC, and looked at the subcellular localization over a few time points. And what you can see here at an early time point, at 10 minutes, you can see that both wild-type and knockout cells are starting to take up the poly-IC through endocytosis, and there is no difference in the punctate area as a surrogate of uptake in these cells. However, at a later time point, you can see that the Sidt2 knockout cells have a significant accumulation of the poly-IC within these punctate structures, which suggesting that the RNA is being trapped in that, or the poly-IC is being trapped. This was also repeated ex vivo, where we took peritoneal cells from our HSV-infected mice, spun them down and stained for the double-stranded RNA. And again, you can see the knockout mice have this accumulation of poly-IC compared to wild-type. And this is quantified here. So these accumulations in the punctate structures we showed were Rab7 positive late endosomes. So that's where Sidt2 was localizing, because if you did the same experiment in MEFs that stably express Rab7 and then treat the cells with rhodomain poly-IC, you can see again the knockout cells have accumulation of poly-IC compared to wild type, and this overlays quite nicely with Rab7 positive late endosomes. This experiment here I really like because it kind of just happened, and I was really lucky to be able to get this result, I guess, because as I was passaging the DC2.4 cells that stably expressed Sidt2M cherry, what I noticed was that you started to get 2 populations later on as you passage these cells. So you get a population of Sidt2M cherry high cells, as well as a population that was Sidt2M cherry low or absent. So using this, we thought instead of throwing out these cells, we could do some experiments with them. So what we did was to treat these cells with poly-IC, rhodamine, as demonstrated in red here. So I've denoted the cells with high Sidt2M cherry with the dotted lines, and Sidt2M cherry low or absent cells with white lines. So we treated these cells, with poly-IC fluorescein, so poly-IC lights up green now. And what you notice is that in the cells that highly express Sidt2, you see less fluorescein signal compared to cells that have low or absent Sidt2. And this is denoted here or quantified here. And this was not simply just due to the Sidt2M cherry low cells not being able to take up the same amount of poly-IC, because you can see that both high and low cells take up the same amount. So we showed that overexpression of Sidt2 was able to enhance endosomal escape of poly-IC. Okay, so if this was true, then our working model was that if you knocked out Sidt2, you would get accumulation of double-stranded RNA within late endosomes, and this leads to impaired signaling through RIG-I and MDA5. If this was true, we would have 2 hypotheses, and the first one was that if you lose Sidt2, you should still be able to signal through TLR3, as shown here, because this shouldn't be dependent on Sidt2, and then you should still be able to get IR3 signaling through the TLR3 arm of the pathway. So to address this, we took our knockout mice again and injected them with either poly-IC, which, as I mentioned, is an analog of double-stranded RNA that is able to hit both TLR3 in the endosomes as well as the cytosolic receptors, RIG-I and MDA5. And then we use a second ligand known as poly-AU, which is specific to TLR3, so it can't stimulate RIG-I and MDA5. So we collected serum after 3 hours post-injection and then measured interferon beta by Eliza again. So in the case of poly-IC, similar to what we saw with the viral infections, the Sidt2 knockout mice produced significantly less interferon beta compared to wild type. However, when you look at poly-AU, you see that the Sidt2 knockout mice are still able to produce interferon beta, and this is predominantly through the TLR3 pathway. Our second hypothesis was that if you lose Sidt2, you would get a loss of RLR signalings within the cytoplasm. If you lose Sidt2, you don't get any detection by regard MDA5, and you knock out this arm of the interferon signaling pathway through MABs. So to do this, to address this further, we decided to measure the activation of the adaptor protein MABs, because that is downstream of regard MDA5, but not TLR3. So to do this, there wasn't a method available for us to use, so we had to develop our own, and we relied on the aggregation of MABs upon its activation. So in these videos, you can see cells that express a YFP MABs construct, either in wild-type cells or in cells that lack the cytosolic receptor MDA5. And then we treated these cells with polyassay rhodamine and watched them over time. And if you focus on the cells around these circles here, as they start to take up the polyassay, they will start to aggregate. The MABs will aggregate along within the cells, as shown here. However, if you have cells that lack the cytosolic receptor MDA5, these cells are incapable of aggregating MABs and activating that pathway, and they're actually just dividing happily. They just don't respond to the polyassay. So using this kind of technique, we took wild-type and Sidt2 knockout MEFs and transfected them with YFP MABs for 24 hours, and then treated them with poly-IC, and then imaged them under confocal. And what we could see here is when you have untreated MEFs expressing YFP MABs, you see this web-like localization, which is indicative of mitochondrial localization. And when you treat the cells with poly-IC, you can see in wild-type cells, they will aggregate in response to stimulation, whereas the Sidt2 knockout mice do not. And this is quantified here over 3 experiments, as demonstrated there. So just to conclude the second part of my talk, I've shown you that the loss of Sidt2 impairs double-stranded RNA endosomal escape into the cytoplasm. The loss of Sidt2 therefore leads to impairment of the MABs-dependent type 1 interfering signaling arm of the pathway, but not the TLR3-dependent signaling through IRF3 and TRIF. So we saw this result, which was great, but then it really led us to ask the question, if Sidt2 is so important in the RLR response, so the cytosolic receptor response, why is that? Because most cells that are infected with virus will be replicating within, the virus will be replicating within the cytoplasm. So the RNA is actually coming from the cytoplasm itself, not through the endosomes. So we wanted to ask, why does Sidt2 have such a profound effect in vivo if these cells are actively making double-stranded RNA within the cytoplasm? And this led us to start thinking about this kind of ongoing arms race between the immune system and the virus. It's known that viruses have developed ways over time to suppress the type 1 interferon response in the cells that they infect to aid in their replication and their propagation. HSV1 is one such virus, which encodes a protein known as US11, which is able to sequester the cytosolic double-stranded RNA in cells that it infects, as well as suppressing RIG-I and MDA5 signaling. And we got a kind of sense for this when we just infected Vero cells, which are cells that are very susceptible to HSV infection. So we infected them with HSV GFP here, so any cell infected will light up green. And then I stained for double-stranded RNA using our antibody, and what I saw was that it was very hard to detect double-stranded RNA within the cells that are green. However, we could readily detect double-stranded RNAs in the neighboring uninfected cells. So this suggested that there was an intercellular transfer of the viral double-stranded RNAs from infected cells to uninfected cells. And this may represent a potential mechanism where the immune system is trying to bypass the viral-mediated suppression of its RLR signaling. So we wanted to formally address whether or not these double-stranded RNAs are in fact being transferred from infected cells to uninfected cells. So what we did was to take Vero cells again and infect them with HSV M. cherry, or leave them uninfected. And then we sorted them after 48 hours, and we got 3 populations. So the first population are uninfected cells, so M. cherry negative, and then you got a population where you have infected cells, which are M. cherry positive, as well as in the same culture, you saw cells that haven't been infected with HSV yet, so with M-cherry negative. So if there was transfer from infected cells to uninfected cells, we should be able to detect viral double-stranded RNA in this population. So to do that, so I denoted them here as either untreated, uninfected, HSV positive, or HSV negative. And then what we did was to do strand-specific RNA-seq to identify whether we can detect viral RNAs within this population and if they were double-stranded. So this bioinformatics work was done by Alex Garnham in bioinformatics. What you can see here is an MDS plot of each population. So you can see clear separation between the infected cells, the HSV positive cells, which are denoted here, and then as well as separation between the HSV negative cells and the uninfected. And they're quite close together in this plot, but if you separate them out, you can see, again, clear separation between the three populations. So then we looked at this further through IGV, and what you can see here is when you look at the first strand and the second strand reads. So up here you can see that the dark blue lines denote a gene along the HSV genome, and then down here you have the reads coming from the uninfected cells. So if you have vero cells that have been uninfected, you hardly get any reads. However, infected cells, so the GFP positive cells, have a large amount of reads across this section of the genome, and you can see that from the first strand and the second strand, they will overlap, suggesting that these RNAs have come off separate strands, but they will form a double-stranded structure. So when you looked at the HSV negative cells, so these are the cells that were in the same culture as the infected cells but haven't been infected yet, you still detect HSV double-stranded RNAs within these cells. And if you look at, again, with the separate strands, you can see that they overlap quite nicely, suggesting that these RNAs would form double-stranded RNA structures. So the next question we asked was, do these viral double-stranded RNAs induce RLR MABs activation? And again, we went back to our microscopy-based quantification method and kind of tweaked it a little bit. So we took MAFs that are wild type or Sidt2 knockout, and we infected them with HSV M-cherry, and we also created stable lines that express YFP MABs here. So every cell will have MABs YFP, and every infected cell will light up red. So we went to Lockie in microscopy, and he devised a quantification method for us based on these plaques or these tile scans. So what he did was to firstly segment out each nuclei based on their DAPI signal, and then I looked at their cell boundaries, and then we were able to number each single cell, and then threshold these cells based on their YFP signal, as well as excluding any cells that were HSV M-cherry positive. So any infected cells were taken out of the analysis. Then what the quantification macro did was to spit out cells one by one to me, and I could score them based on whether they were non-aggregated, so it's still mitochondrial and web-like so. So this is not a non-aggregated cell. Or if there was aggregated, so after activation. And I could just sit there on the couch and kind of just go yes, no, while watching some TV. So I did that for a few hundred cells, and what I saw was that HSV1 infection actually does induce bystander activation of MABs. So what you can see here is an uninfected cell, a wild-type cell. So again, they're web-like and diffuse mitochondrially. However, when you infect them with HSV M-cherry, again, in cells that are infected will aggregate MABs as expected because these cells are, the virus is actively producing double-stranded RNAs within these cells. But you also see, as denoted by the white arrows, that the neighboring cells are also aggregating in response to the infection. So these cells don't have any virus in them, but they're sensing the double-stranded RNA that's being transferred by the neighboring cells. And this is quantified here. What we saw next was that this bystander activation was Sidt2 dependent, because if you did the same experiment in Sidt2 knockout cells, again, you see aggregation in cells that are M-cherry positive or infected, but you do not see this activation of the neighboring bystander cells in the absence of Sidt2. So we took all this data together and we proposed this working model where you have a cell that becomes infected with a virus. The virus replicates within the cytoplasm of the cell and produces double-stranded RNA. At the same time, the virus also encodes for different viral proteins that will either sequester the double-stranded RNA away from the host immune system, or encoding proteins that actually interact with the receptors themselves, and therefore suppresses the innate immune response. When this cell inevitably dies, in the case of a lytic infection, these RNAs are released into the extracellular space and taken up by bystander cells. So these cells are uninfected, but they are taking up the double-stranded RNA from the infected cells. And they take that up through endocytosis, and we believe that Sidt2, in this case, in this scenario, will play a role in transporting the double-stranded RNA across the endosomal membrane into the cytoplasm, and these cells, because they're not infected and they're not being suppressed by the virus, are able to respond to this double-stranded RNA through MAVs and lead to the production of type 1 interferon and pro-inflammatory cytokines. So just some future directions from this work. We have a lot of RNA-seq data to look through now, and we can use this data to identify specifically what are the double-stranded RNAs being transferred from infected cells to bystander cells. And hopefully we'll be able to identify different viral modifications or motifs that are able to interact with Sidt2 and therefore enhance its transport. So has Sidt2 developed recognition of certain modifications or motifs to be able to transport these viral double-stranded RNAs into the cytoplasm? Secondly, we are really interested in what is the mechanism of this intercellular transfer of viral double-stranded RNA. Is it simply just because the cells are lysing and releasing these RNAs into the cytoplasm, or is it something else? There is evidence that suggests that exosomes are able to incorporate viral RNAs and then be released into the extracellular space and mediate bystander activation in this way. So is this happening in our model? Or is it simply just a cell-to-cell contact phenomenon, and it's just cells that are neighboring to each other that are getting these RNAs from infected cells? So a little bit of work left to do, but it's quite exciting to see what comes out of it. And with that, I think it's time to acknowledge the people that have been a part of this work. My supervisors, Ken and Seth, have been a great source of support and education. They've been great mentors to me throughout the last 3.5 years. I owe them a lot. Everything I've learned in the last 3.5 years has been through them. My PhD committee, Mark and Andrew, have always been there to provide additional support and ideas for experiments, and I thank them for that. The information division has been a great place to work, and I love going into the lab every day. I'd like to thank the people there, so especially Ian for welcoming me into his lab. Blake Smith, who we started together working on Sidt2. Mary Lou and Andrew later joined as PhD students. And again, I'd like to thank everyone in the Wicks lab and Masters lab for giving me ideas and taking me out to karaoke nights, because it's been great fun. The bioinformatics analysis was done by Alex in Gordon's lab. Gabrielle Beltz has really helped us with the HSV infection models and just giving advice about viral infections in general. Simon and Greg within the Pellegrini lab were great at the start of my PhD, where they put our mice through all their different models. All the animal techs in room 6, I couldn't have done any of this without you guys. You've been great taking care of our mice and teaching me different techniques and things like that. So imaging has been a major part of this project, and Lockie has been someone who's really driven all the analysis and really helped me make sense of all these images that I've gotten. Everyone in the flow cytometry lab for all their help as well as our collaborators at the Hudson. Michelle Tate, as I mentioned, helped us with the EMCV infections, as well as Kirsten, who helped with the Fret Flim. And Meredith O'Keefe, I didn't show any of the data from that she helped us with, but she's also been helping us trying to get this paper through. And then our collaborators at Harvard as well. And last but not least, family and friends. some of which are here today. I'd like to thank you for all your support, and thank you all for listening. And I'd love to answer any questions you may have. Mark.
Does a Nguyen and MDA5 knockout phenocopy your Sidt2 when you're infected with
EMCV? I know for EMCV, the MDA5 mice phenocopy what we see. So they, again, if you knock out MDA5, which is, as I mentioned, the main receptor for EMCV viral RNAs, all the mice will also die. So we thought that was a good model to follow through with. HSV, it's not as, it doesn't phenocopy as well, but it does seem to play a role. Yep, Tim.
I'm interested in the finding that Sidt2 can recognise a certain length of RNA. I think we've probably talked about it before, but I wonder if there's any indication that viruses, for one, produce different sizes,
yep.
and whether there's some mechanism to they try and avoid detection by producing different size RNA.
Yeah, absolutely. So I think with the RNA-seq data, hopefully we'll be able to start addressing that. Is because you get RNAs coming off from each HSV gene, but you don't know if that's a full-length RNA or if it's because it's a viral replication intermediate. We don't know if it's truncated, the RNAs are truncated, or if it's full-length, but it'd be good to look at that. We also have data to suggest that Sidt2 also recognizes shorter double-stranded RNAs, like sRNAs. So that data that I mentioned before that said Sidt2 can't recognize shorter RNAs, it's kind of contradictory to what we have, but yeah, in our hands, it's able to detect long and short. Actually,
I just want to follow up on Mark's question. So I was wondering whether there was a possibility that Sidt2 could directly activate MABs. And you seem to suggest that the HSV infection in the reguide MD5, hopefully double knockout, don't phenocopy the Sidt2 knockout. So does that suggest that indeed Sidt2 might directly activate MABs, or do you have any evidence to support or refute that possibility?
With the HSV, we know because it's a double-stranded DNA virus, it will also be producing double-stranded DNA, and that can be sensed through the DNA sensors like STING and CGAS and TLR9. So that's why we think the HSV model isn't as important in terms of Sidt2.
So the double-stranded RNA viruses that you've tested behave the same way in a Sidt2 knockout as they do in a reguide MD5 double knockout?
Yes, so the EMCV, if you lose MDA5 in the EMCV infection, the mice will die. Reguide is not that important in the EMCV. Yep.
This is more of a generic question, but if the TRL3 is already there and already inducing the response from the host cell, then why do we need Sidt2?
Yep. So I don't know if I mentioned that as clearly as I should. So it's been shown that MDA5 is more important than TLR3. So it's phenotypically dominant over TLR3. So patients or people with TLR3 mutations, for example, aren't that immunocompromised as you would expect. Whereas if you lose MDA5, you get all this autoinflammatory diseases and things like that. So in that regard, I think the cytosolic receptors are more important in terms of viral infections. We've tried to look at the interplay between TLR3 and Sidt2, and yeah, we don't think TLR3 is playing that much of a role as we initially thought. Yeah. Coffee.
How long did you look at your LCMV infections for, and did you see any differences in anything at all through the experiment?
Yeah, so we looked with the LCMV, if I remember correctly, we looked over 28 days. So it was the more chronic infection, and with the interferon beta results I showed was quite early on. I think it was at day 7 or 24 hours as well. So we did 2 time points, and again, we saw no differences. We assume that the interferon response is quite early on, being an innate response. So we looked at 2 different time points, and again, it was barely detectable. So we think that virus is quite good at shutting off interferon production. Yes?
Why does the poly-IC MDA5 lead to MAVS aggregation?
So poly-IC is a double-stranded RNA ligand, and it's quite long, and MDA5 specifically detects long double-stranded RNAs, so it's able to interact with the poly-IC and signal through MAVS. So once it interacts with, once the poly-IC interacts with MDA5, it'll recruit MAV, it'll interact with MAVS along the mitochondrial membrane, and then that causes its activation.
So MAVS is actually a poly-IC aggregate.
No, so the MAVS will actually aggregate along the poly-IC, I think is the model that people are suggesting. So it'll...
What is MAVS?
MAVS is a adapter protein on the mitochondrial membrane that interacts with MDA5 and RIG-I and causes its, which causes its activation or aggregation. Tash.
Does Sidt2 do the same thing as number 2, and did you just not look at number 1 because it wasn't as expressed as much?
I did not plant this question, but I'm going to answer it anyway. I have slides for that. So if you know me, you will understand that I've had no or very little luck with phenotyping the Sidt1 knockout mice. It doesn't seem to be doing as much as the Sidt2 knockout mice. But we've shown through Blake's work that Sidt1 is also able to interact with, is also localizing within late endosomes and lysosomes, as demonstrated here. And it also, again, interacts with double-stranded RNA or poly-IC with FRET-FLIM, but not double-stranded DNA. So in that regard, it's pretty similar. But one of the first experiments I did was to inject the knockout mice with poly-IC and then read out interferon beta, and it doesn't look like we saw that difference. And given its more restricted expression, it might not be in the cells that are as important in this response. Still don't know what it does. I'm hoping to find out in the last few months that I have here. It's just all going to come together. I think that's what I'm hoping for. Yeah. We do think that it's a inducible gene, so some early results suggested that it upregulates after interferon, but I don't know, as demonstrated here. Does that answer your question? I don't know. Tim.
Just a side over. It's probably a very annoying question, but is it Vero or the cell line that you use to infect? What remind me what it's called?
Vero. Yep, you're right.
How are they immortalized?
They are. So they're a green monkey kidney epithelial cell line.
Do you know how they were immortalized?
No, they're old.
If it was virus, I've always wondered, do you detect levels of, like background levels of virus in those cells because of the previous?
Can't remember how they were immortalized.
It's very
Yeah, they're a very old cell line.
No, did you detect any other background virus, or did you only detect the virus that you...
Really mapped it back to the HSV genome. We haven't mapped it onto anything else. So we could be worth looking into, yeah, for sure. You're welcome. Akshay.
Are there patients who present with mutations with being defective, presumably affected Sidt2?
We do not know, and if you know anyone, please send them our way. I would love to meet them. Thank you, everyone. Oh, Simon, we can talk later.